Hello and welcome to my next video on genetics. Now this is often described as one of the more confusing sections of biology A2, so I hope this all makes sense. Firstly, key terms, there's a lot of them, and I couldn't write them all down, so I've just left key term and I'll explain it. Gene, a sequence of bases on a DNA molecule that codes for a protein. Allele, a different version of the same gene. Genotype, the alleles of an organism has. So for example, if you're writing it down for eye colour, big B is dominant, little b is recessive, that's a genotype, big B, little b. Phenotype, the characteristics the alleles produce. So in that case, big B, little b, you'd have brown eyes. Dominant, an allele whose characteristic appears in the phenotype even when there's one copy. So you have big B, or little b and a big B, it'll still show the big B because it's dominant. Recessive, an allele whose characteristic only appears in the phenotype if two copies are present. It's masked by a dominant gene. That makes sense. Codominant, alleles that are both expressed in the phenotype. Neither one is recessive. So if you have, a white codominant gene and a red codominant gene, you could come out with a pink colour. Locus, the fixed position of a gene on a chromosome, makes sense. Homozygote, an organism that carries two copies of the same allele, so big B, or little b, little b. Heterozygote, an organism that carries two different alleles, big B, little b. Now linkage and sex linkage are the more confusing ones. Now linkage is basically saying two or more genes that are located on the same chromosome are likely to be inherited together. Now why this is? Because it's actually what it's about is the closer two genes are on a chromosome, the more likely they'll be linked together. Because if you've got, let's say you've got a piece of string, two pieces of string, and you're wrapping it over, and you're going to swap them over. One's green, one's blue. If you have two, bits of string that are very close together, two sections of string that are very close together, when you cut a bit off, it is more likely that those two bits will stay together. So if you have two right at the end and you cut a bit off, it's very unlikely that you're going to cut off just one bit of it. You're going to have both little bits right at the end. If though you have one bit, or one little section of string right at the end and one section of string right at the other end, and you then swap half of it over, it's almost a certainty those two are going to be swapped over. And you're not going to have the same two sections on the same bit of string anymore. That's essentially what it is. So if you have two genes on a chromosome, the closer they are together, the more likely they are to be inherited. And sex linkage, a characteristic is sex linked if the gene that codes for it is found on one of the sex chromosomes. So for example, colour blindness, hemophilia, we'll look at a few, usually found on the X chromosome, and we'll look at why that's important a bit later. There's others, muscular dystrophy, fragile X syndrome, vitamin D resistant rickets, there are loads. So the most important tool in the genetics we look at, a Punnett Square. Now I've literally drawn about 10 Punnett Squares in this video, so stay with me. It's going to be quite hard to explain this, but you've got the visual stuff in front of you to look at. So when we look at crossing things over, we have, let's say, a starter generation. They will mate and produce an offspring. That's called the F1 generation. If those generation then reproduce and produce an offspring again, that's the F2 generation, just so you know. And this is how you set up a Punnett Square. This is a very easy one. You have the genotype of the mother and the father. In this case, big N is the father, little n is the mother. You then look at the alleles you have. So in the father, the alleles are big N. In the mother, they're little n. You then draw it into a Punnett Square. So father's got big N on the side, mother, little n. And then it's just like if you're timesing them together. Big N times little n becomes big N, little n. And it's the same for all four. Now, let's explain this one a little bit further. Generally what you'll get is two homozygotes reproducing. In this case, the father, homozygote dominant, mother, homozygote recessive. Now, the dominant gene, the big N, is normal wings. Little n is small wings, so it's got much shorter wings. And then in the F1 generation, all of them will be big N, little n. They'll all have normal wings because the dominant gene masks the recessive. Now F2, this is when you breed big N little n with big N little n, so just that generation, the F1 generation. Now this alleles, you have big N little n from the father, big N little n from the mother. Now if you look at the Punnett Square, you can see that it produces one big N, so two big N's, there's one of them, two big N little n, and one little n little n. Now this means there is a phenotypic ratio of 3 to 1. There are basically saying out of 4 organisms, 3 of them will have normal wings, 1 of them will have short wings. Hope that makes sense. Dihybrid inheritance. Now that last thing was monohybrid. This is dihybrid. There are 2 genes. Now this is slightly more confusing. In the F1 generation, it isn't too bad again because it's always the same thing in the F1. It's homozygotes, NNGG by NNGG. You can obviously see if it's a capital or a lowercase. I'm not going to say big, little all the time. And that's the father and mother. The alleles from the father, big NG, mother, small ng, small ng. If you cross that over, you get all the time big N, little n, big G, little g. And that's for 100% of the population. Now big N's normal wings again. Big G is grey colour in the body. Little n is short wings. Little g is ebony colour. And this is the F1 generation. That one's fine. But then, when you cross them over, you get... So we've got heterozygotes crossing over now. So, the alleles of both the father and the mother, the possible options you have. Because remember, you must always have one N gene and one G gene crossing over. In this case, you can have big N, big G from both the mother and the father. In all these circumstances, both the mother and the father. You can have big N, big G. You can have big N, small g. Small n, big G, small n, small g. And you put it down to a Punnett square. So you've got mother along the top, father along the side. And what you get now is a 9:3:1 ratio. And I'll show you what the actual phenotypes are. But you can see here. So you'll get 1 fully recessive organism, that's the black underline. 3 organisms with recessive N but dominant G, that's red. 3 dominant N's recessive G's, that's blue, and 9 dominant N's and dominant G's. This is the F2 generation. So if we look at what those phenotypes are, now 9 will be normal wings, because that's the dominant one, and grey, because that's the dominant colour. 3 will be normal wings, the dominant wing, and ebony colour, the recessive colour. 3 will be short wings, the recessive wings, and grey colour, the normal colour. And 1 will have short wings and ebony colour, the recessive of both. So that's a Punnett square. Now we can look at a family tree. This is another way of looking at it. This is very good for sex-linked conditions. Now I've actually got some questions here for you to do, but anyway, we're looking at haemophilia A. Haemophilia we do with sex. We do X to the H for haemophilia. X is the female X chromosome, Y is the male Y chromosome. Now, haemophilia, the gene for haemophilia only appears on X chromosomes, so it cannot appear on the Y chromosome, and this is very common with genetic sex-linked diseases. Same with colour blindness. I'm colour blind myself, and for example, well, my grandfather had colour blindness because it appeared on his X chromosome. It doesn't appear on the Y chromosome. If it appears on the X, it means you've got it because it masks out the non-existent gene on the Y chromosome. If you're a female, you must have it on both X's because it's a recessive gene. So all of these sex-linked diseases we look at are recessive. They will instantly become visible if they're on the X chromosome in an XY male. They need 2 copies if they're on both X's in the female. But for me, with colourblind, my grandfather had it because he had it on his X chromosome, but not his Y. Then he had a daughter, my mother, who obviously, since my mother is XX, he must have passed down his X chromosome. Now this made her a carrier because her mum didn't have it, but her dad did. So she's got 1 colour blindness gene and 1 non-colour blindness gene. Since colour blindness is recessive, it masks the effect, so she hasn't got colour blindness, she's fine. Then she had me with my dad. My dad gave the Y chromosome. My mum had a 50-50 chance to give me the X chromosome with the colour blindness on it, and she did, so I'm colourblind. That's kind of how sex-linked disease works. Now in this case, X to the little h is haemophilic. X to the big H is not. So I've shown it here. X little h is affected female, a coloured in black circle. X to the big H, X to the little h is a carrier female. So they've got one gene, they can pass it on, but they don't show any symptoms themselves. And then this is a circle with a dot in. X big H is an unaffected female, no colouring at all, the dot. X little hy is an affected male, that's a coloured in square, and X big HY is an unaffected male. Now I'm going to look at a little more, but let's go through the actual family tree. So it starts off with an unaffected male and a carrier. This person has they have 4 children. Unaffected male, so that means the father passed on the Y, the mother passed on her unaffected X chromosome. Then there's also an affected male, so the mother carried on her affected X chromosome. There's an unaffected female, so dad passed on his X and the mother passed on her unaffected X chromosome. And there's a carrier, so the mother obviously carried on her affected X chromosome. I'm not going to go through the rest of this because you can see it yourself, but what I want you to think about, pause the video and just decide what is the possible conditions of A, B and C. The child's A, B and C. Will they be haemophilic or not, or will they be a carrier? Do it both for men and women. So if it's a man or a woman, what could they be? I'll give you a few seconds to pause and do that. Good. So, A. Now, it's quite hard to actually look at 2 slides at once, but A has an affected female and affected male, so whatever happens, that child will definitely have haemophilia, because the mother must have 2 haemophilic genes. The father has 1 X haemophilic gene, so whatever happens, the mother will pass on 1 haemophilic gene, so the son will definitely have it, and the dad, if it's a female, will pass on his gene that has it, so the daughter will definitely have it. In B, none of the parents have haemophilia or are a carrier, so the child will definitely not have haemophilia. Now in C, the mother is completely fine, but the father has it. Now that means if it's a male, it will not be affected because the father's passing on his Y chromosome, which doesn't have haemophilia, and the mother's passing on 1 of her 2 genes. Neither of them have haemophilia. If it's a female, though, the father is carrying on his X chromosome that has haemophilia, so the woman will be a carrier. She won't show any symptoms, but she will be a carrier. Now this explains why sex-linked diseases are much more common in men. In men, the Y chromosome has nothing to do with the disease, it's only the X. If they have it on the X, they have the disease. Women, they need it on both the X's. So as you've seen there, or in the example, the only way a female can the only way females can actually have haemophilia is if the father is haemophilic and the mother is either a carrier or has the disease herself. But for a son to have it, only the mother needs to have it, haemophilia. It can be a carrier or a completely affected, so a carrier or someone with haemophilia. The father has nothing to do with it because he just contributes to Y. That's why in females, the father and the mother must have the disease or be a carrier. In men, only the mother needs it. So in this case, my mum has colour blindness in a gene, my dad doesn't. That's why I have it. It's much more common in me, but that means I definitely have the disease. Now, if I had a child, there's a 50% chance that my child will have the gene. It's only going to have haemophilia, though, if it's a daughter and my partner has at least 1 haemophilic gene, and even then it's not hugely, like, it's a 50-50 chance. So I hope that makes sense. Now, codominance. Now we've kind of already gone over this very briefly because it's not difficult. In this case, we've got, in cows, CR is red, CW is white. Now, if you have homozygotes again, CR is a red father, CW is a white mother. The alleles are CR, and you can see all the organisms are roan coloured, is what we call it. That's the F1 generation. In the F2 generation, when you have heterozygotes, CR, alleles are CR, it produces 1 CR, that's red, 1 CW, which is white, and 2 CRCW, which are roan. So if you have just normal dominant recessive, in F2 you expect a 3-to-1 ratio, but in codominance you have a 1 to 2-to-1. So codominance isn't too hard, so I hope that makes sense. And now one of the more confusing ones, epistasis. Now what is epistasis? Epistasis is the interaction of different gene loci so that one gene locus masks or suppresses the expression of another gene locus. So you can see how it works here. This is recessive epistasis, by the way. Now, an organism starts off colourless. They need a gene to produce a pigment. This pigment happens to be yellow. They then need a further gene which will then make that yellow pigment red. Without that yellow pigment, the colour won't be red. So you can't have red without the yellow one being produced first. So this is kind of like a little timeline of what happens. You have a colourless albino animal, or plant I think in this case. Now if they have a recessive YY gene, they'll instantly be colourless. It stops there. If they have one dominant Y gene, they will continue on their little path and become yellow. Now if they have recessive R genes, so 2 little r's, they'll stay yellow. If they have one dominant R, they become red. So I've given you some examples here of the genotypes. If they are little y, big R, little r, it doesn't matter that they've got the gene to become red because they haven't got the gene to become yellow in the first place, so they will stay colourless. Big Y, little y, so they will become yellow. Little r, they've got the recessive red gene, so they're yellow. Big Y, so they'll become yellow. But then big R, little r, so then they become red. Now don't think it actually has a timeline. So like when the organism is born, it will start off white, then go yellow, then go red. It's literally, this is just how the genes work. So, you know, at birth, literally it'll be done instantly. That the organism, if it has, let's say, YY, big R, little r, so red, it'll instantly produce that pigment because it's got the right genes to produce it, and it instantly will produce the pigment that'll turn it red. So that's recessive epistasis. Let's have a look at this in a Punnett square. So now I'm doing straight to the F2 generation, not the F1. Big Y, big R, little r times together. The alleles are big R, little y, big R, big Y, little r, little y, little r. You see the Punnett square here. Now I'm going to give an example first. If we had little yr, little yr, you go along, you start off little y, colourless instantly. Doesn't matter about the red, it will be colourless. So what I've done is red, the red coloured pen shows it's going to be a red coloured organism. The blue coloured pen shows it's going to be colourless because I don't have a colourless pen. So you can see here, all of these organisms have little y, so they will be colourless. And then the pink ones will be yellow because yet again yellow pens don't show out very well. So here you can see they have dominant Y's but recessive R's. So the red one is at least one dominant Y and one dominant R. Colourless is no dominant Y's at all, doesn't matter about the R's. And then yellow is dominant Y's and recessive R's. So that's recessive epistasis. Basically, for the gene to mask a gene, it needs to be recessive. So in this case, the colourless gene is masking the yellow pigment. That's how you think of it. So it must be recessive. You also have dominant epistasis. This is the opposite. The dominant gene represses and doesn't show it. So in this case, colourless. If you have one big Y, it will be colourless. And you need 2 little y's to make it yellow. Then if you have one big R, at least one big R, stay yellow, you need 2 little r's to go red. So I've given the 3 same examples I did on recessive, so you can see the difference and one other. First time, little y, little I, big R, little r was colourless because it had double recessive, so it must have stayed colourless. But on dominant, it actually goes on to yellow. And then has one big R, so it goes and stays as yellow. Now, second example, big Y, little y, r. And last time it was yellow, but this time since it's got one big I, it's colourless. Then big Y, big R, little r is colourless again because it's got one dominant Y. Then the only way to make it red is little y, little r. That makes it red. So here is the Punnett square for this. Now, again, the exact same Punnett Square, exact same different genotypes, it's just which ones actually show something. So if I give the same example, now again, blue is colourless, pink is yellow, red is red. So little white R with little white R produces little y, big R, big R. The little y's mean it can be yellow, the big R's mean it will stay yellow. So in this case there's 1 red, 3 yellows and 12 colourless. So in recessive epistasis, the one before, it is much more likely to be the final colour, the final step in the progress. So it's colourless to yellow to red, much more likely to be red. In dominant epistasis, it's much more likely to be the first one, colourless, and more likely to be the one at the end, red. So I hope that makes sense. As I say, this can be very confusing, but I hope this all makes sense. Finally, the chi-squared test. Now, this says, now you're given this formula in the exam and explained what all the symbols mean. Chi-squared, which is x squared, equals sideways m, which is the sum of O minus E squared over E. O, where O is the observed values and E is the expected values. So what does this mean? What is this actually doing? This experiment is there to test the null hypothesis. The null hypothesis means that in genetics, or not in just genetics, but in stuff where you have chance, anything which is off the expected value is just down to chance and not an actual reason. And the example I'm going to use here is flipping a coin. Flipping a coin should be 50-50. Now in this case you've got 48 tails, 52 heads. Now we're going to test to see if that is just down to chance or if the coin is like fiddled, it's heavier on one side. Now I can tell you now, if it was like 30 tails and 70 heads, then yes, the chances are it's probably, there's something wrong with the coin rather than it being down to chance, because it should be 50-50. Obviously only 100 flips isn't, as perfect as we want. We would want, thousands of flips to make it actually scientifically accurate. How we test this is you'll get a table. I suggest you look in your book on page 135. There's a little table. It'll show the number of degrees of freedom, and what you want to look at is the critical value, which is at 0.05 or 5% probability that the deviation is due to chance alone. You then find out the chi-squared value, which we'll look at in a minute. Compare it with your value in the table. If your value is less, the chi-squared value is less than the value in the table, then it is down to chance. If it is bigger than that value, then something is actually happening. There's a reason to it. Now, before I actually go into numbers, degree of freedom, what does that mean? It's degree of freedom is a number of outcomes minus 1. In this case, tails and heads are the two outcomes, 1 degree of freedom. If you had, for example, in the epistasis, for example, you're looking at chi-squared there, you have 3 classes. You have colourless, yellow and red, so that there are 2 degrees of freedom. That's how it works. And so I've told you, if you look at the table, please while you're watching this look at the table, it makes a bit more sense. At 0.05 or 5%, the critical value is 3.84. So for our flipping of a coin to be 48-52, we want a chi-squared value of less than 3.84. Hope that makes sense. So here's my table. Expected value is 50-50 for tails and heads. Observed value is 48-52. Now we do observed minus expected. 48 minus 50 is minus 2. 52 minus 50 is 2. We then square that value. Minus 2 squared is 4. 2 squared is 4. We then divide that value by the expected value. So 4 divided by 50 in both cases, and that's 0.08. We then add those two values together, 0.16. That's our value for chi-squared. 0.16 is less than 3.84, so the null hypothesis is correct, and this is down to chance. So I hope that makes sense. I say this is very confusing and it's very hard to explain over a video because I can't actually point things out or draw things on. So if you need to rewatch it with the book open, to let you know, a lot of the time I'm using the CGP revision guide, if that helps, for the examples. Anyway, conclusion. You have monohybrid, that is just one gene. Dihybrid, 2 genes. You have dominant genes which mask all genes, recessive genes which must appear homozygous to actually be shown, codominant where both genes have an equal effect, epistasis, which is when one gene can mask another gene which will then go on to do something else. It's kind of like a chain of genes. Genetic diagrams, we had the Punnett Square, which I showed you originally, which is more useful, and the family tree, which is useful for sex-linked conditions and diseases. And you can test whether your genetic problems are down to chance or not by the chi-squared test. So for example, in the population, if you had 1,000 people, 750 of them were brown eyes and 250 were blue, you'd know that you can do that test and work out that actually you've got a perfect sample. Anyway, thank you for watching. Leave any comments, likes, or email me, and goodbye.